I'm trying to change my name.
Great. So, good afternoon to everyone and welcome to another Connecting Three Worlds seminar. Here at Connecting Three Worlds, we explore the health cultures produced by socialism across Africa, the Americas, Asia, and Europe during the Cold War. So please find more information about the project in our website. My name is Sebastián Fonseca. I am a postdoctoral fellow at the project's Latin American team. And today we are very pleased to bring together three fantastic scholars to have a conversation about the history and genealogy of Cuban biomedicine during the Cold War. We welcome Ms. Fatima Elfitouri from King's College London, Nils Graber from the University of Lausanne in Switzerland, and our very own Professor Dr. Edna Suarez from the Universidad Nacional Autónoma de México in Mexico. As the moderator of the session today, I will first explain the format of the seminar to our viewers, and then we will begin. Each guest has 15 minutes to present their work around our main topic today. I will first formally introduce each scholar right before their presentation and then cue them to start. The order for today will be Fatima, then Nils, and finally Edna, and we will end with questions and a conversation amongst ourselves. My aim is to keep within the time limit, so I will signal them on the video or raising the hand option in the Zoom, and if needed, just a quick intervention by activating my microphone. So having said that, let us begin. Our first speaker is Fatima Elfitouri, currently a last year PhD candidate at King's College London in UK with a project titled Cancer Immunotherapy in the Cuban Bioscape. Fatima's research focuses on the development of cancer immunotherapy and biotech in Cuba. She conducted ethnographic research at the Center of Molecular Immunology at La Habana, Cuba, where she will be speaking from. Prior to her doctoral work, she completed a BA in cell biology at the University of Manchester and an MA in medical anthropology at SOAS University. Fatima has also conducted biomedical research on cancer at the University of Nottingham and has worked to research on anti-malarial resistance program at the MRC unit in Gambia. We thank you again for being with us, and the floor is yours.
Thank you. I will share my screen. You have to give me, can you make me a co-host? Thank you. Can you see my screen?
Nope.
No? Okay.
There we go.
Now?
Now we're good. Yes, now we have.
Great. So let's see. Okay, so today I really wanted to, I actually thought this was more of a, I guess, an informal session to kind of discuss our research and kind of the thoughts that we had. And I'm working on an article right now, and I thought I'd present it to you and get your thoughts, and within it kind of explore my own, I guess, the thesis and kind of what I'm doing with it. And so the title is Curative Logic in Immunotherapy in Cuba. And I think I should begin by situating myself as in why did I actually pick immunotherapy and Cuba specifically? And this has a lot to do with, so I studied, as Sebastian mentioned, cell biology, molecular biology as an undergrad, and I had an opportunity to work for a year at the Medical Research Council in the Gambia, and I was working in a lab conducting research on antimalarial resistance. And it was quite fascinating, kind of the sense of incongruence between kind of the education that I had about how science is conducted, what science allows us to, or how science rather allows us to discover things about the natural world, and the objectivity discourse. And when I was in the Gambia, it was so evident and clear to me that funding, politics, and culture influence scientific production from the beginning all the way to the end. And so I really wanted to kind of explore this relationship between science and culture, science and politics in more depth. It was very important for me to have a different image of science in the global south, because as it stood for me at that point, and Gambia reinforced that, is that science was mainly conducted or funded by the global north. And I wanted a really different narrative that of course exists, but I really wanted to experience that and see that firsthand. And so I came across really through my reading about immunotherapy in Cuba and how they developed cancer vaccines. And I was shocked because I'd really not come across cancer immunology in my education or in my research. So I really wanted to delve in this and explore it further. So I'll begin, I guess, by giving a brief history, and it will make sense why I'm beginning at this point. But one of the narratives when it comes to biotech and science in Cuba generally begins in the 1960s, and I do follow that genealogy, but I thought for this session, I'd maybe step back a little bit and begin maybe at the turn of the 20th century, when Cuba just emerged as a new republic after it gained independence from Spain, and kind of the role of US medical interventionist policies and how they affected and shaped the development of public health. So at the turn of the century, physicians and political elites really believed in science. And this actually began from the 19th century, if not earlier, that science really was the way for the nation to develop, for the people to develop, and for them to gain independency and maintain political sovereignty. And so when kind of with the US interventionist policies that were really aimed at controlling the spread of yellow fever from the island, from Cuba to its southern ports, so there were US interventions that were trying to control that, and what these physicians and political leaders did was try to use those interventions to kind of build a public health sector, and to have this, and that they had this view of, I guess, medical nationalism, as Rodriguez writes in this wonderful book called The Right to Live in Health. But by the 1930s, their view of health was really challenged, because it was based on ideas of kind of producing a modern subject, kind of through this notions of hygienic modernity. But this was really rejected by many factions, from feminists, from journalists, who argued and demanded that health and disease should be centered within kind of the racialized and gendered neo-colonial economy, and it could not be just seen as a biological element or a biological phenomenon, but rather it was social, it was political. And so really it's at that point that social medicine begins to form in the 1930s and develops, and it's picked up again in the 1960s and is made into, I guess, what public health today looks like in Cuba, kind of the socialist public health. I'll move on to the next slide. So when the revolution took place, which took place in 1959, the next decade saw kind of tremendous investment and change in development in knowledge policies. So in 1961, you had the literacy campaign, which about 250,000 volunteers, mainly young women, were sent across the island to educate and teach people how to read and write. And the other issue that the state had to, or the revolutionary state had to deal with, which is was to repopulate the professional strata, because after the revolution, many people left. So you had doctors migrating, and just many of the professionals left. And so the state was under tremendous pressure to replenish that population. So it nationalized that wasn't, I guess, the main reason, but I guess part of the consequences. So yeah, part of what it did was really try to democratize knowledge and access to knowledge. And so it sent people abroad, the literacy campaign and a whole host of other campaigns. And it had a number of policies throughout the years, and I focus on two science and technology policies that it developed. So from 61 to 77, really the kind of science and technology policy was supply oriented. And it was all about the promotion and development of the research and development sector. By the late 1970s, another model began to take shape, which is the central control model. So while the first really focused or had this understanding, quite a simplistic idea of investing in, I guess, developing the research and development sector, but it had this idea that if it invested in basic science, then industry would come and use that knowledge and develop technology that would then translate into better welfare and kind of greater economic growth, but it didn't quite work out. So they changed their, I guess, their orientation and developed this kind of centralized control model that really gave birth to the biotech industry. And the centralized control model was really relied on having a network of collaboration and wanted to join everything together. So from basic research to manufacturing and kind of the different aspects that moved knowledge into something that could be used and utilized. So the central model, what it did by the late 1970s began to so what the state did, it invested a lot of money from the 1970s and 80s onwards into creating these polos scientificals or research production centers. And these centers, each one was based on a particular technology. So the first one that was developed was the CIGB, which was based on recombinant DNA technology using bacteria. And later, in 1992, at the height of an economic crisis, the Center of Molecular Immunology was completed. And all these centers were built on a closed cycle model. So they were fully integrated centers. So you had research, production, development, marketing, all under the same administrative unit. But the closed cycle model has two, I guess, layers or two facets. You have the vertical, which is from production to marketing. And then you have the horizontal, which is the relationship between biotech centers and public health. And often I think, from my interlocutors, public health or biotech is seen as an extension of public health, which is an important feature, I think, in Cuban biotech. So. I can't see the. Sorry. So to just give, I guess, more context of the, so SIM was, as I said, was built in 1992, but the group that would later occupy SIM were based in the oncological hospital in Havana. And they were working mainly with chemotherapy, but then in the late 70s, a new technology develops, which is hybridoma, which is a way of developing or a way of producing monoclonal antibodies. And this kind of revives, in some way, immunotherapy, because immunotherapy had many, I guess, it had risen since the, I guess, late 19th century, and it would, like every few, I guess, every decade or so, it would become popular and then it would wane, like the interest in it would wane and then it would come up again. So you had a huge moment in the 1970s with interferon, but by the time these Cuban scientists get involved with immunotherapy, it had fallen out of fashion. So they come in with hybridoma and they begin to develop and, I guess, work with this therapy. They had lots of problems like transferring antibodies into the clinic. And they had, but then they just, I guess, in their development, they encountered a lot of problems. But just, I guess, a brief history of immunotherapy, which currently stands as the fifth modality of cancer treatment, together with surgery, radiotherapy, chemotherapy and precision or personalized medicine. And it started with quite primitive, in one way, kind of techniques where, the example of cholestoxin, where this doctor would inject toxins to activate the immune system in the hopes that by stimulating the immune system, that the immune system would get rid of any cancer in the body. There were also resistance studies where people would inject tumors into patients, again, to stimulate the immune system. But by the 1970s, with BCG and interferon, kind of this magic bullet moment for cancer appears, where there's this hope and hype that that's it. We really understand what cancer is about, and we have all these advanced biotechnological tools. We can cure cancer once and for all. But that, of course, falls flat on its face. But then, of course, again, it's revived in the 21st century with CAR T-cell therapies, where T cells from patients are taken, re-engineered, and injected back into the body. And then you have, of course, checkpoint inhibitors, where kind of function in different ways, but still this kind of curative idea that cancer can be managed and can be managed through biotechnological intervention. So I think you should ignore this slide, because I figured that Nils would really delve into this much more than I can here. But I just wanted to mention that what happened in Cuba is that when these antibodies and immunotherapies begin to move into the clinic, the pharmacokinetic dynamics and the way they operate in the clinic is quite distinct from chemotherapies. And so the scientists have kind of an issue, because these immunotherapies are not working in the way that chemotherapy is supposed to work, which is it's supposed to shrink the tumor very quickly. It's the way it's given is really based on kind of the optimal schedule is really based, not the optimal schedule. It's the way it's given is based on the maximal tolerated dose. But they find out that immunotherapies don't work in that way. And so they begin to rethink kind of the logics, the curative logics, and the models, the immune system, or the immunological models that these immunotherapies work on, and how it actually differs from infectious disease. And so I'm really struggling with this technological. Anyway, so what they do as they have this divergence is that they suggest that we need new analogies to think about what is going on with these immunotherapies. And their main point is that with chemotherapy, the logic that's being perpetuated is one of infectious disease, one of really getting rid of the pathogen, one of complete cure. And what they argue is that cancer really, potentially there are some cures, but the majority of cases, or at least, so one thing that I should mention is that they really focus on advanced disease, and they treat them kind of distinctly in some way. And so the center, their main focus really is on advanced disease, because they think a lot about chronicity and the way that people can live with cancer in a way that is manageable. So they problematize this idea of antibiotics, and of that is really a legacy of the bacteriological revolution. And I think I should also, talk a little bit about that. And it's worthwhile just giving, again, a brief history that, in the 19th century, you had the discovery of infectious agents, and later, just a couple of decades later, people begin to work on how the body really responds to these pathogens. And they come up with these models that are based on defense. So, for example, one of the forefathers of immunology, Eli Michnikov, in his lab, one day he observes, so what he does is that he has a bunch of starfish in his lab, and he injects one of them with a rose thorn or something. And what he finds out is that a bunch of cells come and begin to kind of eat away at this intruding object. And so he first of all names these cells phagocytes, eating cells, and he argues that this is the body's way of, this is kind of the defensive mechanism of the body, and that the body is in its nature a defensive organism. And that is a fair point, but kind of looking closer at it and looking closer at the term immunity, I think quite a bit with the scholar Ed Cohen, and he wrote this wonderful book called A Body Worth Defending. And in it, he argues that defense as a way to understand what the body does is really it's a political and a legal term that emerges. The history of immunity itself, it moves from legal to political and then to immunological, and it carries all this baggage with it. And so immunity as a term is not clean, or it's not devoid of a lot of baggage. And so he really asks us to reconsider and to think about how we, like the terms that we pick and the work that they do in our understanding of the world. So there's this concept of defense and how limited it is. But if we fast forward 50 or 60 years later, different models of the immune system emerge beyond its defensive posture. And people begin, this actually happens really early, but it just doesn't get picked up or is not really represented. And I think this remains to be the case, where the defensive model, the militaristic, it's still very powerful. And we've seen it kind of with the pandemic, where these divisions still remain. But what these scientists offer is that they offer different models of understanding the immune system. And so they use things like danger theory, and they use autoimmunity to rethink what the immune system does. And one of the things that they argue and really try to place at the forefront is this concept of tolerance, and the concept that the division between the self and the non-self that has previously been understood as established early in development. What they argue is that it's really in continuous motion, like this distinction between what the cell is and its demarcation from the outside is one under contestation, and that immunotherapy could really engage in that. And it's a continuous process that evolves with the body. I think I'm out of time, but I guess I just wanted to wrap up by saying that I guess my main argument that I wanted to kind of present here is that the way, first of all, the movement of immunotherapeutic products from the lab to the clinic and back again through this closed cycle, as well as the biological results that has come out from the clinic, but also the way biotech is seen as an extension of public health creates couplings that maybe are not seen elsewhere of the molecular and the molar. In that, one of the most kind of prominent thing in my own, in my fieldwork was kind of this mention of the patient and how scientists who are really far away from the patient talk about the patient. And you see kind of this linking between, cell biology or molecular biology with public health, with something much grander. And I will stop there. Thank you very much. And sorry for going over time.
That's fine. Thank you, Fatima, so much for that. We will obviously speak about your work more towards the end of the conversation, but thank you so much for that. Okay, so up next is Dr. Nils Graber. He is a senior researcher at the Science and Technology Studies Laboratory located at the Institute of Social Sciences of the University of Lausanne in Switzerland. As a medical anthropologist, his recent work is focused on immuno-oncology and precision medicine at the SNSF project DOFIS, which explores the development of personalized health in Switzerland. Just very recently, actually published an article on the idea of chronicity in medical practice and technology. Very interesting piece, worth checking, and obviously connects with what Fatima was just talking about a minute ago. He has also studied cancer research and biomedical innovation in the primary healthcare system in Cuba. And Nils has graduated from the doctoral program in social sciences and health at the School of Advanced Studies in Social Sciences in Paris. So again, thank you so much for joining us, and you can take it away.
Thank you, Sebastian. I'm very glad to participate to this conversation. Can you see my screen well? Yeah, okay. So I hope my presentation will be not too long. I thought it was 20 minutes, so tell me when I'm about 10 minutes just to know if I need to accelerate or skip a
No
few
problem,
slides.
don't worry, you're good.
And so I decided to focus this talk on one chapter of my thesis, one of the most historical, let's say, and related to the cooperation between the Cuban scientists, clinicians, and the Second World. So through a cooperative group based in the former Comecon. And so it will be complementary to Fatima's talk and more related to the history of this field of immuno-oncology in Cuba and the tensions between clinicians and laboratory researchers. So I will be quite quick with the Center of Molecular Immunology, which was one of my main fieldwork. I just wanted to emphasize here the joint venture agreements of SIM with other countries.
Sorry, Nils, I think yes. I need to stop because the presentation is not in the PowerPoint, the full slide, so we can't see you go forward or backwards. So I don't know if you can, if you've done that already. I think we skipped it.
Sorry, what is the problem? You have the small version of the PowerPoint?
It's only the small version, so we can't see whether you're moving forward or backwards.
And here? I tried to change. No?
I don't know if the other ones.
Here.
Yeah, I don't know. Does Fatima and Edna, can you see the slides in full?
I will just use one screen in that case. I think it should work with this. How is it now?
I think it's still. Oh,
I will share again. Sorry, just. What do you have?
I think you need to flip because you're using two screens, so we can see sort of the other side, the one that is showing you, the slides that come up next, but not the actual full screen.
Yeah, I'll try again. Sorry for this.
That's all right, don't worry.
So, how is it now?
Same thing, but you know what? If you can see it going through from slide to slide, then we can see it as well, and it works fine. There we go. Now we can see. Now we can see it. Now it's good.
I had to plug off the other screen. So yeah, just wanted to emphasize these joint venture agreements of SIM permitting to develop part of the innovation trajectory with other countries, until now mostly with China, Thailand, and in the past with Brazil, Argentina, India, and quite recently with the USA. And it's also a way to enhance the capacity to export these pharmaceuticals. So here I'm questioning the form of oncology at stake in Cuba by using cancer immunotherapy. in existing literature. I'm building on the work of Keating and Combrosio, describing oncology as a global style of practice, which has been shaped by transnational networks such as cooperative groups to organize multi-site clinical trials and define new guidelines. So this transnational standardization of the field. And also the fact that over the last 3 decades, genomics and more recently cancer immunotherapy have been reconfiguring the knowledge and norms of oncology. Furthermore, in existing literature, oncology has been described mostly in relation to the global north, while in the global south, it has been characterized mostly by forms of improvisation of medicine, if we follow Jolie Livingston, for instance. So my fieldwork consisted of interviews with clinicians, researchers, regulatory authorities in Cuba, and also an ethnography of a clinical trial of cancer immunotherapy that took place in primary care settings as an experiment, not only of the treatments, but of the whole organization of care with this public health vision that Fatima emphasized. My fieldwork took place between 2014 and 2017, but I'm still collaborating with Cuban scientists and researchers in relation to these primary care clinical trials. I also analyzed scientific publications, gray literature, and a few archives related to international collaboration. Nevertheless, in relation to the second world, I will build mostly on interviews, so let's say oral history, and a few textual fragments, mostly publications that I found. The emergence of the field of and the industry of cancer immunotherapy, and I will be very fast with this because Fatima explained it before. So it was envisioned as a lever of development and emancipation from the sugar-based economy. It was developed from scratch by relying on this long-standing experience in tropical medicine and the early scientific development in the 60s. And also, and I would like to emphasize this, the fact that the biotechnology industry was constituted by establishing a kind of biomedical elite in Cuba, receiving direct state support with a political mandate to develop at the same time economic value and public health value. So the first biopharmaceuticals developed in the 80s were interferons, monoclonal antibodies, and prophylactic vaccines. Interferon was heralded as a wanted drug in the 70s and 80s globally, as a pleiotropic drug, to take the word from the historian Troy Peters, because it could serve as an immunotherapy of both infectious and chronic diseases, including cancer. So in Cuba, this interferon story started with exchange between the US and Cuban scientists in 1980 under the mandate of Jimmy Carter, who wanted to normalize the relation between the US and the communist island. So Fidel Castro decided to send Cuban scientists in Houston, Texas at a cancer institute and in Helsinki to learn how to produce interferon. And when they came back, they received the full support from the leader Maximo to reproduce what they learned abroad and to produce locally a leukocyte interferon. And we see here a few photography of this. And I quoted this website of Bohemia, a culture journal in Cuba, who wrote a beautiful piece about the story of interferon, of course, quite laudatory about the role of Fidel Castro, but nevertheless, we see very nice pictures of the scientific facilities at that time. And the first leukocyte interferon was used in 1981 against hemorrhagic dengue epidemic outbreak, and a bit later against cancer and hepatitis. In 1983, some Cuban scientists could be trained at Pasteur Institute in Paris to learn about the recombinant way to make interferon by cultivating bacteria. And this is the biotech process that was implemented at the CIGB, the Center of Genetic Engineering and Biotechnology, so the biggest biotech center in Cuba, established in 1986. And in parallel, in the 80s, monoclonal antibodies were starting to be produced at the National Institute of Oncology and Radiobiology, the main cancer hospital in Cuba. We see a picture here on the right. As interferon, monoclonal antibodies was heralded at that time as magic bullets, permitting to make diagnostic tools and treatments with exquisite specificity, to quote Cambrazo and Keating book. So the production started at the Department of Immunology at INOR in the early 80s. And biochemists at INOR were also studying cell surface markers such as CD6 and EGFR, which is one of the key targets of the Cuban immunotherapies. First clinical applications were undertaken at the end of the 80s, and the investigation at INOR did constitute the basis for the creation of SIM in the early 90s. In parallel to this, oncologists at INOR were actually already involved in conducting clinical trials of chemotherapy. And they started with the leadership of the director of INOR at that time, Zoilo Marinello Vidarreta, who was trained in the US in the 40s, and then was among the only 11 oncologists who stayed in Cuba after the revolution. So he was a highly committed politically member of the Central Committee of the Communist Party in Cuba. He was also a member of as a foreign member of the Soviet Academia of Science, and the representative of Cuba within the Technico-Scientific Committee at Comecon. So he tried to involve collaboration with both Western Europe mostly and the communist bloc. And when he established in the early 70s a department of experimental chemotherapy, he tried to promote clinical trials in Cuba by integrating the Comecon or the CMEA cooperative treatment group that was centralized in Budapest, Hungary, since 1978. It was created before in 1973, and this cooperative group was mostly experimenting chemotherapies produced in the Eastern Bloc. The clinical trials, the multinational clinical trials undertaken within this group were mostly phase 1 and 2 protocols against breast, ovary, and head and neck tumors. So we see here a very interesting cartography of global oncology involving Cuba, but also most of the Soviet countries and the Eastern European countries member of the Comecon. From Dushanbe to Havana, we can witness how multinational clinical trials were undertaken. And here I'm taking an illustration from an article I found in the Czechoslovakian scientific journal, Neoplasia, about comparative clinical trials of different combination of chemotherapies. So here I'm quoting a Cuban oncologist who told me during my fieldwork that for her, the first experiment of domestic biotechnologies such as interferon against cancer were highly problematic. She told me, With interferons, the first steps were very blind. Leukocyte interferon was used. The first response was obtained against myeloid leukemia. Interferon was also used against renal cell carcinoma, but it was an experiment as well. It was not very notable. There was a search for antibody formation, but at that time, they were not well regulated at CIGB with the technology of phase 1 clinical trial. At INOR, we were shocked to see how things were done without methodology. So we see how this tension between laboratory and clinical settings was taking place in the 80s when the biotechnology elite had the power to undertake clinical trials without taking stock of the experience of clinicians in Cuba who were trained with the Comecon cooperative group. So now my last part about the post-Soviet clinical trials. So the CMEA or Comecon cooperative group disappeared in 1989. It was replaced by a Central and East European oncology group, that still exists today and collaborate mostly with the US and Western Europe to participate to multi-site clinical trials. So in Cuba, clinical trials had to be relocalized to a certain, mostly because this cooperative group disappeared and the Soviet Union collapsed, so they had to undertake the first clinical trials of cancer immunotherapy, mostly at home within the island. To accompany this, new infrastructures for the regulation and organization of clinical trials were established for this. The Drug Agency in 1989, the SECMED, and the National Center for the Coordination of Clinical Trials in 1992. In the early 90s, so CIM had to undertake clinical trials of both monoclonal antibodies and therapeutic cancer vaccines, which was a concept at that time, I don't want to deepen, but we could talk about it later, that was chosen as an alternative to monoclonal antibodies, because when these mAbs were made of murine cells, they generate toxic effects. So that's why at that time, before the capacity to humanize monoclonal antibodies, they decide to generate an immune response against EGF rather than targeting the EGFR with a monoclonal antibody. So these are two of the main products of CIM today. And one of the CIM immunologists told me that for him, the severe crisis of the 90s, called the special period in time of peace, was an opportunity to strengthen the link between the laboratory and clinical settings. So he told me, here comes the disaster of the 90s. So this context was at the same time favorable in terms of how the research and our immunotherapy products gain rapid access to clinical practice. Oncologists realized that there was an option that practically in many situations, what it had to offer to some patients, especially very advanced, were the products that we were doing at CIM, because there were huge shortages at that time of chemotherapy, for instance. So you were saying oncologists at that time were very, at the world level, quite reticent to the theme of immunotherapy, but that was not the case of Cuban oncologists, because to a certain extent, they had only the same products to offer to advanced cancer patients. So that's how a new paradigm could be shaped all of the 90s. Following global trends in biopharmaceuticals, CIM's researchers argued for a new paradigm of clinical evaluation, partly related to the epistemic specificity of local immunotherapy. For instance, they consider immunogenicity as a surrogate marker and not the diminution of the tumor size, for instance, which was a surrogate marker for chemotherapy before. Furthermore, long-term response rather than short-term one, so the ability to diminish the tumor size in the short term was not considered as a main endpoint because of the own rhythmicity of the immune system to generate a specific immune response against a tumor antigen. So thanks to more tumor-specific drug, chronicity becomes a therapeutic goal focusing on diseases stabilization rather than eradication of malignant cells, as Fatima explained. So the phase 1 clinical trials were undertaken at the end of the 90s until 2006 for the publication. The phase 2 clinical trial of the therapeutic vaccine I'm referring to was published in 2008, and the phase 3 in 2016 in well-known oncology journal. But CIM had difficulty to undertake all of the phases of clinical trials locally in an island of 11 million inhabitants. It took a lot of time to finish the phase 3 clinical trials that ended in 2016. That's why in the meantime, it attempted to, through joint venture agreements, to offshore some part of clinical trials, especially the phase 3. And now that's the case with the recent US-Cuban collaboration taking place at the Roswell Park Center in New York. And they started in 2016, a first clinical trial of a Cuban cancer vaccine in New York. So it was approved by the FDA as a drug to administer, as an experimental drug to administer to US patients. And this clinical trial associates the CIMAVAX EGF, the Cuban vaccine, with a blockbuster immunotherapy called nivolumab. So it's also a way to position the Cuban drug within a highly competitive market, and also a way to accelerate the development of these drugs and to get access to other tests that are not available in Cuba, especially in genomics. And CIM scientists also try to keep participation of Cuban hospitals to transnational clinical trials. And during my fieldwork, there was a transnational clinical trial sponsored by the Singaporean firm Inokiz, which involved different hospitals in Southeast Asia and Inor. But the oncologists told me that it was very difficult for them to participate to such a high-level first world clinical trial, because it's extra work for Cuban oncologists. They are not paid for this specifically, and they have to keep their normal practice along with the clinical trial. So at the end, I just wanted to make this last point about the regulation in Cuba of these biopharmaceuticals. Building on both the experience of the domestic biotech industry and global regulations, the Cuban Drug Agency segment has developed a specific regulatory environment, allowing CIM a triple strategy of clinical trials. In the cases of large-scale clinical trials, they try to completely offshore them. In the case of multinational clinical trials, they try to keep their participation, as I said. And at the same time, they try to undertake large phase 4 clinical trials in primary care to enhance public health impact and focus on chronicity management by virtually giving access to all lung cancer patients today in Cuba to the therapeutic cancer vaccine, and to produce population data in terms of impact at the population level. However, because no approval has been obtained until now for Cuban drugs in the global north, and because of the avalanche of new drugs in oncology in the global north, Cuban oncologists tend to consider local immunotherapies in primary care, as many told me, as kind of borderline. So in conclusion, transnational clinical trials of chemotherapy with the second world, but also biotechnology cooperation with the first world, have both been crucial pre-existing experiences for the development of cancer immunotherapy in Cuba. The post-Soviet crisis has fostered cooperation between bench and bedside in a time of relocalization due to economic constraints and isolation. Local Cuban oncologists have to negotiate different kinds of norms, which continues to generate tensions with biotechnology experts. What we can call the state biotechnology regulation of biopharmaceuticals permits to maintain different kinds of clinical trials in Cuba, aligning global and/or local norms, including expanded use within primary care. So I will end here. Sorry for being a bit long.
That's all right. Thank you so much, Nils. Right. So finally, our last speaker is Professor Dr. Edna Suarez. She is one of our principal investigators here at Connecting Three Worlds, coordinating the Latin American team. Her work in the project focuses on the medical genetics, technical assistance, and interventions in Latin America and Mexico during the Cold War, specifically the connection between human disease and the studies of human variation as it played out in the decades after World War II. Her previous work on the topic covers the 1960s studies on a range of blood diseases, for instance, hemoglobinopathies in Mexican indigenous communities, which took place in the context of the Malaria Global Health Campaign. She has extensively worked on issues around evolutionary biology, the introduction of statistical analysis in the life sciences, and the mobilization of nuclear sciences and technology in Latin America. She is currently the Dean of the Department of Biology at UNAM, and without further ado, you can start your presentation. Thanks. I think you have to unmute yourself just on the side. And we can't hear you. No. There we go. Yep. Now we can hear you. Yep. That's all right.
So yes. Okay. So well, maybe I should start also like Fatima and Nils saying a little bit about my trajectory and why I'm here, because I'm a completely newcomer to the history of public health and global health. And basically I come from the history of science, and I have been moving towards biomedicine, the history of biomedicine. So because of my subject, and you will see that, I mean, your two talks are so interesting for me, and they are, I think we have a lot of connections. For instance, one of the things that Fatima mentioned was this fact that, you know, we had this impression that not only funding, but all the research, the interesting research on biomedicine and basic science that related to medicine happens in the global north. And of course, we know that this is not the case, and that there is a lot of research, what we will call basic research in biomedicine connected to public health, that also takes place in other places. So basically, I've been studying the Cold War period, and I'm going to talk a little bit about my previous work, and actually this is a very baby project that I'm starting, and you will see why I want to. I think the Cuban connection will be one of the many connections that I want to do with this history. And my talk today has to do, and this project that I'm just starting to think about has to do with a very, how can I say, a universal technique or technology in many laboratories of molecular biology, which is electrophoresis. And so I will, let me see, how can I change? Okay, so, but no, let me see, is that I cannot read, and I didn't take the precaution of printing my my presentation. I wanted to show you this picture of Ruben Lisker, a geneticist, a very just passed away recently. And he was a medical geneticist, and his team in the hospital of the Institute of Nutritional Research in Mexico did a lot of research on blood diseases. And because of this research, we have a very complete of one of the most important surveys of genetic traits in indigenous populations in Mexico. So in the past, I have worked a lot on Lisker's work because of the many connections that he enables us to think. So I'm going to talk a little bit about, I'm going to start by reading an anecdote that is important for the project that I'm thinking about. So in 1960, after returning from his graduate studies in Chicago, medical geneticist Ruben Lisker was granted 1,000 Mexican pesos grant, amounting to less than $100 US, to start a precarious research project on blood types and abnormal hemoglobin in indigenous populations. He also received a small amount from the Mexican Secretaría de Salud, the Health Ministry, and the local Carnot laboratories. But the meager funds did not stop him to pursue the kind of edgy research that he had become acquainted with at the Michael Reese Hospital in Chicago, Illinois. With the help of a friend, engineer Mauricio Rusek, he used part of the money to build a small apparatus of paper electrophoresis, one of the new options to perform the decades-old technique of separating macromolecules, proteins, by their electric properties and size. At the hardware store, they bought and adapted a pair of glass plates, put together an electric power supply, a couple of platinum electrodes, and forceps to hold everything together. With this made-up instrument, Lisker managed to perform the first electrophoretic studies of genetic variation in Mexican populations. His main concern was to identify the same abnormal hemoglobin he had worked with at Chicago, sickle cell hemoglobin, or HBS for short, as well as the presence of G6PD, glucose 6-phosphate dehydrogenase deficiency, a scarcely understood but prevalent mutation in African and Mediterranean populations. A few studies had been performed before on abnormal hemoglobin in Mexico by many others, which had not been successful. Nevertheless, in collaboration with his partners at the Hematology Department of the Instituto Nacional de la Nutrición, Lisker soon published the first of several papers, actually 19 papers, on the genetic variation of local populations, reporting several genetic variants in blood proteins. Though he continued to use the old serological test and blood typing surveys, his first report of a G6PD anomaly made use of electrophoresis to describe the clinical case of a Jewish Sephardi boy whose family had immigrated from Turkey to Mexico. Later, using the technical variant of starch gel electrophoresis, he reported the presence of abnormal hemoglobins, including a Chiapas variant, in Mexico's indigenous communities. Lisker's research, which continued until the late 1960s, remains to this date as one of the most extended genetic surveys of Mexican populations. So I wanted to tell you about this anecdote of Lisker doing this made-up instrument with things that he just bought at the hardware store, because electrophoresis has a lot of interesting features. That I mean, it's a molecular technique of the new kind of analysis of macromolecules. It has been very important in the discovery of and the establishment of many important facts, not only in molecular biology, but also in evolutionary biology, in biomedicine, and nevertheless is still and remains a very cheap technology if you compare it with other kinds of technologies like, I don't know, like ultracentrifuge, of electron microscopes, or today's DNA sequencers. So this speaks a lot about the mobility of these technologies. And in particular, the analysis of blood diseases got a very quick start, especially in the 60s, using electrophoresis because of its close relation with the global campaigns on malaria eradication that, as you know, were very important for the World Health Organization. In 1956, Mexico became the first country to sign up on the global campaign to eradicate malaria. And very soon, I mean, even during the war, the hypothesis that blood diseases, and in particular sickle cell anemia, were related to immunity to malaria infection had been one hypothesis that had been used by not only by physicians, but by evolutionary biologists to explain why there was such a global spread of sickle cell anemia in populations that were very close to malaria infection. So if you compare these couple of figures that I'm showing you in this presentation are just among many examples where you see how the two geographies, the geographical distribution of malaria presence and sickle cell anemia always overlap. So, I mean, it's not a coincidence that the hypothesis was first formulated in studies by the British Empire on the African colonies. And since then, it has been one of the main examples in evolutionary biology of how a mutation that can be had very negative consequences on African populations can nevertheless be remain into the present in the populations and not being eradicated by natural selection, because as you know, if one person is not homozygous for this mutation, they can still be immune to malaria infection. So it provides a certain immunity. But very soon, people doing this service, especially in the 50s, they started to unveil other different variations in blood proteins. And one of them was a very complex group of diseases called the thalassemias, which are mostly a disease, a molecular disease related to the regulation of production of the different chains of hemoglobin. So the thalassemias are a kind of genetic regulatory disease at the molecular level. And then they soon also realized the presence of this very common anomaly. Actually, G6PD is the most common genetic variation among global populations. So it's the most prevailing type of genetic anomaly or abnormality, as the physicians say. And all these diseases also overlapped with the malaria distribution maps. So they thought that there might be some kind of immunity provided. But the interesting case of G6PD anomaly was that physicians in Chicago in the very early 1950s realized that people carrying the G6PD anomaly were prone to terrible hemorrhages if they ingested some of the anti-malarial drugs that were used to fight malaria. So in this case, it became very important to know which person had this mutation, because it could have a very bad reaction with Primakin and other of the anti-malarial drugs. So it's very interesting because this blood disease, which is G6PD, it's also connected to favism in Middle East countries, and it became the first genetic disease for which there could be a pharmaceutical consequences. So it's been hailed as the origins of pharmacogenetics, not pharmacogenomics. But in the 50s, it was the first way. Well, so in the past, I've been studying what I call the blood diseases in the backyard. And by that, I mean the very interesting networks of collaboration between physicians in the southern part of the United States, where malaria was still prevalent. And of course, sickle cell anemia is prevalent among the African-American population, which lives in the southern parts of the United States, and the malaria and blood diseases prevalent in the coast, in the Gulf of Mexico and the Pacific coast of Mexico. So it's very interesting because the geography of the disease has to do with the distribution of poverty and questions of race. Okay, so this is only one of the tables of one of the many scientific committees organized at the World Health Organization, in which physicians from all over the world, and of course many physicians from so-called third world countries at the moment, gather to keep information and data about the common distribution and the data on the malaria hypothesis, meaning they confer immunity by all these anomalies to these populations living in these areas. So there was a lot of investigation of these blood diseases, and what is very interesting is that many of these genetic studies, as I say, happened because there were a lot of resources devoted to the malaria campaigns, and but then they started to look at all these problems with some of these drugs that they could in some African American populations and African populations, as you can see this in the description, they saw that there was evidence for this very strong reaction that, of course, was put a limit on the use of these drugs into these places. So that was my project until recently, but because of this new project in which we are trying to kind of rewrite some of the histories of biomedicine and global health from the perspective of socialist countries and socialist contexts and socialist internationalism, we have started to see, to look at some of these relations. And one of the very interesting things is that very soon, in the 50s and especially in the 60s, a revolution took place with electrophoresis. It became much more cheaper than it used to be. There were a number of technology firms that started to produce this equipment, and also all the consumables that come with electrophoresis. So for instance, some media which are already prepared for scientists to be used in many places were standardized, so the scientists didn't have to devote a lot of time preparing all these things. And what is interesting is that there were many networks of geneticists, physicians and geneticists that did this research, but around the world. For instance, one of the most interesting characters in my story is James E. Bowman, who was an African-American doctor who studied in the United States. Eventually became the first African-American to hold tenure in a Chicago hospital and be admitted after the Civil Rights Bill was passed in the 60s. But before that, and after returning from his service in the military, in the US Army, he had to get a job in the Middle East. So eventually he traveled to Iran, and he did a lot of research on blood diseases and G6PD. And when he was able to return to the United States and get a job in this hospital in Chicago, he became the director of the blood bank in Chicago. And he was very important, for instance, in genetic surveys of Vietnamese indigenous populations, which were very, very important during the Vietnam War. So you can see how this the study of blood diseases, because of the importance for malaria in context, or especially of war, for instance, was also kind of had lots of priorities into the US agenda, and especially in the 60s and in the 70s with the rise of civil rights and segregation, etc. But the thing in my project is that many of these technologies, even though they were produced in the United States, were also used and imported to places like the Soviet Union, like Czechoslovakia, to other places. So I really want to start exploring this. Okay, Sebastian, I'm about to finish. This is today's price. As you can see, is not a very expensive technology, and I wanted to finish. Is my on to the last. This is the electrophoresis in my brother's lab. He's a molecular biologist today, so you can see that everybody, he says, everybody has the same, like in Chile, like if you go to Japan, like if you go to the UK, everybody has the same equipment. So of course, the questions that I'm trying to answer with this project have to do with the global mobility and the use of technologies in context of poverty. And of course, here I'm following the historian of technology, David Edgerton's plea to do this kind of history. Of course, one interesting thing, and for that I would like to interview and get a lot of research on Eastern Europe, is the mobility of not only techno-scientific practices, but of these technologies across political borders. Of course, questions of race and ideology are already into this project. The historian of science, Elise Kevorkton, for instance, has studied the history of the geneticists in the Middle East, and how the study of these blood variants is completely related to issues of nationhood. So that's also very interesting topic. And of course, the relation between biomedicine and global public healthcare, because as Fatima was asking, in reality, many of these geneticists, for instance, in Mexico, in Colombia, in the Middle East, were not only gathering data for explanations and basic scientific research in the global north, but they were also trying to provide explanations for mechanisms, for causes, doing the kind of basic science that was incorporated in lots of global health campaigns. So basically, this is the project, and with that, I finish.
Wonderful. Thank you, Edna, and thank you everyone again for their presentations. I suppose we enter the last phase of our little meeting. Yes, it was more of an informal conversation. I just wanted to have a bit of a structure so that we had more clarity on how to move forward. But the idea is to talk about sort of all the common topics, all the common themes that you can see cut across. Now, I want to open basically the floor for anyone to just ask a few questions and stuff, but perhaps one of the themes that I can see connecting every single one of our conversations have to do with the paradigm of infectious diseases that was very strong throughout the Cold War, the global health history during the Cold War. It seems to me that this is one of the topics that sort of connects the three conversations together. So perhaps there's some space to think about that going forward. But I can see, obviously, Edna has her hand up to start igniting the conversation. So I'll let her speak and take it from there.
Yeah, no, I was dying to ask you a couple of questions to you all, because it's so fascinating what you're doing. Of course, well, as you can imagine, also I want to do this research on these blood diseases in the Cuban context. So that's why this is one of the places that we would like to explore in the project, of course. And it's just fascinating that you have done so much. And Nils, you say that you're not a historian, but nevertheless, I mean, your project covers so much. I wanted to ask you a very simple historiographic question, but before that, I wanted to mention to you that one of the postdocs in our project, Lu Chen, she's doing a project on Chinese biomedicine, and she's going to present a paper at the 4S meeting in Cholula about how not only the clinical trials, which of course in China they don't have the problem of a small population, but the theoretical background of biostatistics was kind of dramatically cut off after China split from the Soviet Union. So she will talk about the difficulties of the Chinese pharmaceutical and biotech industry, all the challenges that they face once they could, with this theoretical background from from kind of universal or not universal, kind of global developments in biostatistics that were very difficult for the Chinese. The other thing that I think it's very important for you too is Dora Varga, who is also a PI in our project. She wants to study the connection between the Eastern European countries, especially Yugoslavia, Czechoslovakia, Hungary, with Cuban doctors, and also how they came to Cuba just after the revolution, when many of the physicians, as Nils was telling us, migrated to the United States. And I wanted to make a very maybe trivial and mundane question about your sources, because when you read in the general media about Cuban biomedicine, I mean, and you read about these Cuban doctors or scientists talking about their research, it's very, I mean, there is a kind of mythology about the importance, for instance, and you mentioned this of Fidel Castro or the revolution, and all this talk is part of the way in which they describe their developments. So I would like to know your thoughts on that. What are the difficulties for you? What are the limits for interviews? And or how did you go through this kind of narratives to get to other interesting points? Yeah.
Nils, do you want to take this?
Well, I think it's for both of us.
I mean, you can begin.
So up to you if you want to start. Or...
Yeah, I think it was one of the things I noticed when it came to Fidel Castro and the scientists that I interviewed is that there was this generational break, in that with the older generation, Castro was huge. He, was the face of everything. Although there was, of course, for example, when I interviewed people like Agustin Laje, who was the first director of SIM, he really put himself back and presented this story where Fidel Castro was doing everything, although he was a major political and scientific player and remains to be so. Maybe not political so much, but at least a scientific player. But with the younger generation, Castro was not part of really the conversation. He was a figure from a different time, and so they were able to talk, and they were very critical as well. I think the younger generation were very critical of the science, of how science was managed and is managed, and I think particularly the narratives that were presented by the state about the embargo and other things. I mean, people were, and we've seen with the recent revolutions in Cuba, prior to the pandemic, they were, you really felt it, that people were really on edge, and the pandemic allowed people to express lots of ideas that they had. So yeah, I really saw this kind of disjuncture between the two generations, or the different generations.
Yes, I would agree with this. And furthermore, well, I think that in the biotechnology field, the discourse of Fidelistas is still strong, perhaps less in the younger generation, but at least during my fieldwork, it was still stronger than in hospitals. So you also have this quite of privileged situation, and still today, I think, in terms of salaries, in terms of social protection. For instance, now in the crisis, people working in the biotech industry, they receive goods directly at their work. Nevertheless, there have been some criticism inside of it because of the bureaucracy, because of who will govern this industry. And that's true to some extent, that Fidel Castro, to a certain extent, let's say, managed to control the bureaucracies and ministries, and to involve this direct relation with the biotech field that was kind of out of the political infrastructure, and in direct relation with central committee of the Communist Party. And to some extent, it's true that at least in the biotech, in the health biotechnology, Fidel Castro had this gesture of delegating the power to the biotech leaders. And actually, one of the director of the biotech of the whole industry was José Miguel Barrueco, who was a physician. So the hand of the physicians of the profession was really strong until the 8th of the 90s. And with the change of direction of the generation, there is this critique of bureaucracy that has emerged, and there is a famous article, I think, in Nature Biotechnology of a former director, Limonta, Manuel Limonta, I think, who was head of CIGB and left to the US, and who wrote a quite critical paper about the organization, the risk of bureaucracy, of skipping, of diminishing basic science for the sake of exportation and commercial relations. So my answer in terms of sources would be that as anthropologists, we try to multiple the sources. So not only, I mean, in terms of the professions, I think it's important in my case to interview the clinicians as well, but also people who left Cuba, I think, provide interesting and contrasting voice, definitely. And then the foreign researchers who established collaboration with Cuba, for me it was key as well. French scientists, Swiss scientists, and definitely they have a different version, more critical, at least when they are not in Cuba, but in the laboratory in Europe. Yeah.
Wonderful. I also wanted to ask just very quickly, I'm very interested on the international connections that are being made during this time that you guys spoke about. Obviously, Nils mentioned the case of Zoilo Marinello, kind of producing that connection with the European CMEA. But I was also wondering if maybe Fatima has found other connections. Obviously, I've worked also with Latin American social medicine, which has a branch that worked with Cuba, and I met some people from Cuban biomedical sciences that studied at the State University of Minsk during the 1970s, part of a state program of cooperation and technical collaborations between the USSR and Cuba. But I wonder if there's anything else that ought to be considered in the development of Cuban biomedicine that is centered around the networking that happened at the time. And I wanted to know just how much impact that those connections made in what you guys have studied on immunotherapy and such.
So you're talking about these international connections that are based on social medicine. So, I mean, my main sources really was STS scholars in Cuba. I mean, they really helped orient me in thinking about, I guess, how science was imagined in relation to medicine and to public health. And they had lots of connections with French scholars, but also with Latin American scholars. But this was mainly to do with kind of science and technology, and kind of looking at the relationship between science, technology, and society, and how to kind of move forward and utilize or enhance that relationship. But when it came to social medicine, there really, I haven't.
No, sorry. What I mentioned just social medicine because some of the scholars there studied basic sciences before getting into social medicine, but the conversation is about biomedicine. So I'm talking more about those connections in biomedicine.
Yeah. So it's. I'm actually. I don't think I got your question. Can you repeat it again, or I'm misunderstanding somehow?
No, just I'm wondering if you guys could expand on the networks, on the international networks that happen around biomedicine with other countries in the USSR or the Soviet bloc and Cuba, or any other connections that were quite critical in the development of biomedicine in Cuba. So, for instance, Nils mentioned Marinello. And so sort of expanding that a bit more, I find that fascinating and interesting to look at.
So, in my work, I guess one of the things that I really wanted to do was look at Cuba and not focus, although people have tried to divert me to look at these international networks, but yes, I think Nils has done more of that, but I really wanted to tell, I guess, a version or the development of immunotherapy by really focusing on what the Cuban scientists were doing, as opposed to, I felt that it was a slippery slope for me to look at the international context, because then it would always be, oh, they had to go abroad to learn, and everything kind of came from abroad. And so I really just wanted to focus on what was going. And of course, it's really impossible because it's a very, I guess, international network of scientists. Most of them studied abroad. They have lots of collaborations abroad, and even kind of these new movements of kind of having venture or, like inviting, I guess, hybrid. What they often would do, for example, when scientists would travel abroad to complete their PhDs or maybe do a couple of postdocs abroad, they would try to have joint patents on their inventions so they can move these products further, because, for example, SIM could only invest in a couple a year. And so they really encourage students and junior researchers to kind of establish links between, with other universities to be able to kind of progress their product, even though they only have kind of partial control over it, but it was better than kind of the alternative. So it's very important that they continue to do this. I think one institution that was really interesting that I did not expect was I think it's the Weizmann Institute. Weizmann Institute in Israel. There seems to be a lot of connections with people coming in. I first met these researchers at a conference that SIM organized in 2018, the biennial conference. And lots of people coming from all over the world. So I think that's maybe a really good place to look at these relations, international relations, because not only did, in these meetings, not only did you have scientists come, but you had lots of people who are strictly in biotech and in the private sector coming in, which was kind of interesting to see, lots of Americans traveling illegally to the US, and kind of the stories that you hear. But the other thing that I guess I should stress is the absence, who wasn't present in these meetings is just as crucial. So I only attended one meeting, but I did ask a lot about who actually has been present before and after. And there's kind of the lack of, most people were from Europe and the US. And I was surprised that there weren't as many or really any Latin American scholars or African scholars. And so it's kind of interesting kind of how Cuba positions itself, it does something in certain cases. But I guess the argument was that this is where the resources are, and we're looking for resources. We're looking to expand and we need this. And so it wasn't about, creating collaborations for a greater purpose, but it was really about, it sometimes felt about survival and expanding the market.
Right. Yeah. Yeah. Nils, if you want to add something, or Edna, you can also chip in whenever.
Yeah, I think this network has a lot to do with oncology, and if you look at other public health issues, and even in biotechnology, I'm sure you will find more networks with Latin America, Africa. But it's true that in the 70s and 80s in oncology, it was really the collaboration with Western Europe and the former socialist bloc that was really key, as I try to emphasize, and the USA, that depending on their government and willingness to approach Cuba, and as Obama did more recently with this joint venture with Roswell Park, it's all of this diplomacy to also, which is quite fascinating to consider that this US diplomacy is looking at Cuban innovation as potentially very interesting drugs for their own system, and in that case to establish exception to the embargo. But in terms of technology transfer, I think that's interesting to look at. It's a bit more recent, but definitely the technology of monoclonal antibodies, for instance. I think that Cuba did play a key role to transfer this technology to Brazil, for instance. I don't know about Mexico, but with Brazil for therapeutic maps and Fiocruz, I would like to start interviews with them, but apparently the initial platform came from Cuba, and it's interesting in terms of doing biotech with lower resources, in the sense that they rely on their own reagents, for instance, their own way of transforming bioreactors from academia to industry, and then to lower costs. So they have a whole package of biotech for lower resource context that is very interesting to look at. I think for India as well, the first monoclonal antibodies stem from Cuban platforms.
Yeah, and I mean, you're mentioning also in the chat the Centro Nacional de Investigación Científica.
Yeah, that was more for you, the period that you may be interested in, the 60s, 70s, 80s. I think that was a key institution for more basic science and also biomedicine. In Cuba, the CENIC, which does still exist, and most of the leading researchers in biotech stem from this institution.
Worth looking at the archives. Edna. Oh,
can I just add something, because it's what we're mentioning, is that there's kind of strong connections between Cuba and China, and that there's been this technological and institutional, like the whole closed cycle model has been transferred in this biotech institution based in China. It would be really fascinating to kind of explore further.
Wonderful.
Okay, so I'm sorry, Fatima, did I understand well? I mean, this full cycle model was kind of exported from Cuba to China, not the other way around. So that's really fascinating. Yeah, very, very successful model. I mean, there are so many interesting things, because it's really amazing how this little island has become so important in terms of many of the questions that we're asking about biomedicine, and especially during the pandemics, for instance. You know, this the use that has been for a long time, has been going for a long time, of medical diplomacy and the exportation, the distribution of vaccines. I forgot to mention, and this could be also interesting for you, one of the collaborators of the associated with the project, but of course he has huge research projects by himself, is Maciel Yavi, who is working on the politics of drugs. He has work on that, and he's now going to travel, and he will do research on the connection between Iran and Cuba, which is also kind of very interesting. I just wanted to kind of start to close our seminar, because we have so many things in common, but one thing that really is for me, it's very important is this the development of clinical trials and the restraints it has put on the pharmaceutical products and the implementation of these protocols, etc. And this is really interesting because not only because I mentioned that, of course, as more biostatistic requirements have been, growing, that now, for instance, you not only need to do these trials in a given population, but you also have to have a sample of ethnicities or race, and we see the retooling of race into kind of pharmacogenomics. But as I mentioned, it was also important for the development of pharmacogenetics. And once I was talking, I forgot the name of this researcher, but how places like Mexico, for instance, who is so close to the United States, and for Cuba, has a huge population of course, Hispanic population, which is very representative of the own Hispanic population in other places like the United States. So it has become an offshore for doing this kind of clinical trials. So it has developed, it has become like a kind of secondary market for collaborations or place of collaboration, that it seems that maybe something very specific about it should be written about the kind of, the restraints it puts and also the possibilities for networking. So with this, I think I could finish, and we need to keep with our conversations. That's what I would say.
Yeah, thank you, Edna. I also have so many questions about these new paradigms that were set in the progress of immunotherapy and such, but I do think it is all the time that we have today. I will stop now the recording, but before I end, just a last thank you for being here, and yeah, I wish you all the best in the work.
Thank you.